Hello, everybody, and welcome to the Canadian Lightsource STEM Seminar Series. I would like to welcome and introduce you to my friend and colleague, Joel Reid. He is an industry scientist with our team here, and I'm going to ask you to introduce yourself.
All right. So my name is Joel Reid. I work in the industrial services group here at the c.l.s. My background is in engineering physics. I have an undergraduate in engineering physics, and I also have a PhD in engineering physics. But it was very much a mixture of disciplines, some material science, some chemistry, some physics, a little bit of everything, a little bit of biology too, because I was working on synthetic bone when I did my grad work.
I remember that.
Um, c.l.s is a dream job for me. I tell all of my friends that I'm doing one of my top two job choices in Canada, and the Maple Leafs won't return my calls. So yeah,
Awesome.
I absolutely love being here.
Great.
With that, I guess can I start the presentation?
Sure. I just like to get started in a good way and offer to folks to identify where we are. So we are located on the prairies, in the heart of the prairies. We are in Treaty 6 territory, which is traditional homelands of the Nakota, Dakota, Lakota, Anishinaabe, and Nehiyawak First Nations. It's also traditional homeland of the Métis peoples. And we are walking in our education team, we are walking a path of reconciliation where we recognize and honor the traditions and knowledge of the people who walk this land before us and with whom we share. So with that, Joel, please go ahead and share your screen.
All right. So I'm just gonna share my screen here. Yeah, so today I'm going to talk a little bit about what a synchrotron is and what we can do with it. And I'm really pleased to speak with you today. So it's really important to state up front, I am a scientist. And what it means to be a scientist really depends a little bit on your perspective. In my job, I try to help academics and industrial clients find answers to scientific questions that would be very hard to answer without using a synchrotron. I want to try to genuinely convey some science without using any equations today or any math, just some images and pictures to keep things understandable for everyone. And hopefully the big picture will make sense. I'm going to start by giving you a brief introduction to synchrotrons in general, the Canadian Light Source specifically, and the industrial services group, which I belong to. Then we'll take a look at some capabilities of the c.l.s, specifically concentrating on two categories of techniques, including diffraction and imaging. I'm going to use a lot of short examples to illustrate a range of different applications. And early on, I'm going to go into a few little detours where I explain concepts or background in more depth and hopefully answer some of your questions as they come up. So the first question is, what is a synchrotron? A synchrotron is a particle accelerator, and it creates very intense beams of X-rays to study materials. To create the X-rays, we first create a beam of electrons using a device called an electron gun. Electrons are tiny negatively charged particles, and they're normally attached to atoms. The electron gun uses high temperature and high voltage to separate the electrons from atoms and accelerate them into a beam. This beam of electrons is transferred into a booster ring, where they're further accelerated very, very close to the speed of light. Finally, the beam of electrons is transferred into a storage ring, where they circulate around this ring continuously. The storage ring has experimental end stations that are called beamlines coming off the ring tangentially. Every beamline has a magnetic element, and this magnetic element is used to bend the electron beam. This magnetic bending gives rise to synchrotron radiation, or very intense beams of X-rays. Individual beamlines are designed to make optimal use of different techniques and different energies. Ultimately, really, a synchrotron is a materials analysis tool with many different capabilities. So the next question is, what are X-rays? X-rays are a form of high energy radiation. This figure illustrates different types of radiation, and as you go from left to right in the figure, the energy of the radiation gets higher. So X-rays are a form of radiation with higher energy than visible light, which everyone should be familiar with. And you can see all the forms of electromagnetic radiation here and some familiar sources of each type of radiation at the top. One of the interesting things about radiation is that all radiation has a wavelength, and that wavelength gets shorter as the energy gets higher. The wavelength of X-rays are comparable to the size of atoms, so X-rays are very good probes for studying atoms. X-rays are perplexing because they behave like both particles and waves. So you need to think of them in different ways in order to explain different types of behavior. If they are not scattered by matter, X-rays travel in a straight path. And with an appropriate detector, you can actually count individual X-rays, like particles. Individual X-rays are called photons. But since X-rays have a wavelength, they can undergo what are called interference effects, which are more like waves. Interference is the interaction between two or more waves. In this case, we're talking about X-rays, but the most obvious everyday example is waves in water. If you've seen overlapping waves created when two boats pass, you'll understand constructive interference. So constructive interference occurs when wave peaks and wave troughs are perfectly aligned between two waves. This results in a wave that's twice as big, like the one you see on the left here. When waves are completely misaligned, you get what's called destructive interference, and the two waves cancel each other out, which is illustrated on the right. So constructive and destructive interference apply to X-rays in the same way they apply to waves in water. And we're going to come back to this concept shortly. So a synchrotron is a particle accelerator, and it creates really intense X-rays for studying materials. Synchrotrons typically have multiple experimental stations called beamlines, and each beamline is designed to perform a specific technique. This is a typical outline of a beamline. Beamlines generally have three components. There is a primary optical hutch, which is used to focus the beam and to select the wavelength or energy for your experiment. There is an experimental hutch with the samples and the detectors where the actual experiment takes place. And finally, there's a control area nearby for running the experiment. And we use synchrotrons to look at the structure of materials. So an important question might be, what do we mean by structure? When we study the structure of materials, we often need to look at different length scales in order to understand different aspects of their performance. So the example shown here at the top right is what's called a battery pouch cell. This is a commercial battery format that gets used for research quite frequently. The figure below is a computed tomography, or a CT image, and it's illustrating the layered electrode structure inside the battery where you store all the energy. Structure like this, that's on the scale of millimeters, is what you would call macrostructure. If we zoom in 100 to 1,000 times, we can see the individual electrode layers inside the battery and even the individual particles that make up these layers. We call this structure microstructure, and there are 1,000 micrometers in 1 millimeter. If we go another 1,000 times smaller, we're on the length scale of atoms that make up the material, and we call this structure nanostructure or atomic structure. And structure at all these different length scales can be really important for explaining how this battery works. We just mentioned atomic structure, so we should probably review what elements and atoms are. Elements are unique pure substances that combine together to make up all of the matter that's around us. Atoms are the smallest building blocks of these elements. So atoms themselves are made up of three types of particles: protons with a positive charge, neutrons that don't have any charge at all, and electrons with a negative charge. I've shown three really common elements here with the number of protons and neutrons and electrons in their atoms. Over 90% of our body mass is actually made up of these three elements. Most of the time, when we illustrate atoms and structures, we use colored spheres to represent the atoms, like what I've used in the top row here. The main takeaway on the slide is that the elements are unique pure substances, and atoms are their smallest building blocks. When you combine different elements together, the substances are called compounds. Here are two compounds most people are familiar with: water, or H2O, and carbon dioxide, or CO2. The atomic arrangements for these two compounds are called molecules. So this illustrates one molecule of water and one molecule of carbon dioxide. The compound on the right is the lithium battery material, illustrated a couple of slides previously. This compound arranges in a structure of repeating layers, and it's what we would call a crystal structure. We're going to look at this concept of crystal structures a little more closely in a couple of minutes. Right now, we are looking straight down the layers, but if we tilt the structure slightly, you can see the three-dimensional nature of the structure. So structure is really important because it is intimately linked to the properties of the material and the synthesis method, or the way the material is made. When we design and develop new materials, we're generally trying to obtain very specific properties from these materials. For instance, when we design metals and alloys, we are typically looking for high strength and durability. And when we design pharmaceuticals, we're trying to target specific health issues and diseases. We're also trying to synthesize them in ways that are repeatable, cost-effective, and easy to scale up for production. Synthesis properties and structure materials are all interrelated. So generally, if you want to understand why a material works the way it does, the first step is to determine and understand that material's structure. We were on this slide already, and we took a little detour to explain what we meant by structure and why structure is important. So we use synchrotrons to study structure of materials used in a wide variety of different disciplines and areas. I've listed some of these areas here, but let's take a little bit of a closer look. Here's a sampling of different areas where synchrotrons are used. This is not a comprehensive list of different applications, but what I've tried to do is show a broad range of different areas where synchrotrons get used. We won't be able to look at all of these areas today, but we will look at examples from a number of them, including pharmaceuticals, energy materials, minerals, and agriculture, among others, as we go. Let's take a little look at our synchrotron in Saskatoon. So I work at the Canadian Light Source, which is Canada's only synchrotron facility. Construction of the c.l.s. began in about 1999, almost 25 years ago. And the c.l.s. has been operating since about 2005. But many of our beamlines were built more recently or have received significant upgrades. We have about 250 to 300 employees, and these range from scientists and engineers and technicians to finance and human resource specialists. We are used for everything from fundamental research conducted by academics at universities to very applied research for industrial clients. And industry access is a core mandate of the c.l.s, and it has been since the conception of the facility. So personally, I work in the Industrial Services Group, or the ISG, at the c.l.s. We predominantly work with companies, but we also help academics to access the c.l.s. for fee-for-service work. And one of the privileges of our group is we get to work with numerous beamlines and techniques, looking at a wide range of different types of samples. We are very fortunate to get to participate in many different areas of research. There are a number of ways to access the c.l.s. Academics who have excellent researcher ideas and are comfortable waiting for beam time typically use General User Proposal, or GUP access. We normally have 2 calls for proposals a year where proposals are graded by peer review and the best proposals receive beam time. So for successful proposals, it generally takes about 6 months to receive your beam time. Some researchers prefer guaranteed access without peer review. With purchase access, you don't have to worry about peer review or publishing your results. The users run their own experiments, and there is an expectation that they have synchrotron experience. For non-experts with sufficient funding who are just interested in the results, full service is an option. We typically do this for companies, but occasionally we perform small feasibility studies for academics as well. And for experts who just need data quickly, we have mail-in services. The c.l.s has both paid mail-in services and mail-in services that are related to GUP access. This is a floor plan of the c.l.s. The c.l.s is a relatively small synchrotron facility, but we have a fairly wide range of different techniques, and everything from infrared radiation, which is quite low energy, to very high energy X-rays used for scattering and computed tomography. I've specifically pointed out the 2 infrared beamlines here in red. All of the other beamlines at the c.l.s use X-rays for their experiments. And I'm not going to go into any detailed technical specifications for individual beamlines. We could end up being here all day. But as I go through examples, I will try to highlight some of the beamlines that are being used. So there are 3 main categories of synchrotron techniques, including diffraction, imaging, and spectroscopy. I'm going to give you a basic introduction to 2 of these techniques, X-ray diffraction and X-ray imaging. Synchrotron techniques in general tend to be very fast and element specific with higher resolution than equivalent lab techniques. And this means that you can actually see more details. This also makes them very well suited to time-resolved studies, where we track changes in the material while we perform the experiment. And many of the advantages of the synchrotron also lend themselves to hybrid techniques, which kind of blur the lines between these 3 categories. So we're going to start with a technique called X-ray diffraction. Before we look at any specific technique, let's consider what a basic synchrotron measurement actually looks like. If you visualize a synchrotron measurement with a simple schematic, the first step is we shine X-rays on our sample. Then X-rays come out of the sample, and we measure those X-rays with some type of detector. The X-rays that come out of the sample tell us something about the structure of the sample. This is really all a synchrotron measurement is. X-rays in, X-rays out, where the X-rays coming out provide information about the sample. For our first technique, X-ray diffraction, when we focus X-rays on the sample, the X-rays are scattered by the sample. The patterns these scattered X-rays make tell us about the atomic structure of the sample, specifically the way the atoms are arranged in what are called crystals. So before we go any further, what exactly is a crystal? Crystal is just like a brick wall made up of identical bricks. If you know what one brick looks like, then you know how to build the entire wall because the pattern repeats in 2 dimensions and every brick is identical. A crystal is also built from a repeating pattern, but the pattern repeats in 3 dimensions instead of in 2 dimensions. In a crystal structure, instead of building with bricks, we build with something called a unit cell. The unit cell is the basic building block of the crystal, just like a brick in a wall. This is a unit cell for diamond, which is made up of carbon atoms. This unit cell itself is made up of 8 smaller blocks, 4 with a tetrahedron of carbon atoms in them, and 4 that are basically empty. When you know the arrangement of the atoms in the unit cell, you can build the entire crystal. You just stack up unit cells in 3 dimensions, just like bricks in a wall. When you stack up millions of these unit cells together, we get a diamond crystal. There are a couple of different techniques we use to study crystals, depending on the size of the crystal. When we have a large crystal that's easily separated, so that we can look at a single isolated crystal, we use a technique called single crystal diffraction. Large can be a relative measurement. You can see the mineral crystals on the left here on the order of millimeters in size. These protein crystals in the middle figure range from about 20 to 200 micrometers in size, and they are all very suitable for single crystal diffraction. When crystals are very small, like the top right figure here, on the order of 1 micrometer in size or smaller, we have to look at many thousands or millions of crystals at the same time using a technique called powder diffraction. And we're going to take a look at both single crystal and powder diffraction here as we go. Here's what a single crystal diffraction experiment looks like. So the crystal is mounted on a metal base like this, that's roughly an inch in height. To actually see the crystal, we need to zoom in on the top of the base. And the crystal is this small cube in the center of this loop. This crystal is on the order of 0.1 millimeters or 100 micrometers in size. So it's not very big. When we focus X-rays on the crystal, we get a pattern of spots on our detector. This is a cartoon pattern, but an actual pattern looks something like this. The pattern depends on the orientation of the crystal. So we collect patterns of spots at many different crystal orientations by rotating the crystal in small steps and collecting a new pattern at each step. Typically, we collect hundreds of 2D patterns from a single crystal. Computers are then used to analyze the 2D patterns and to determine how the atoms are arranged in the crystal. A few minutes ago, we talked about how X-rays behave like both particles and waves. We looked at interference and how waves that are perfectly aligned can undergo constructive interference. This is essentially what diffraction is. Every dark spot you see in the 2D pattern on the right is a case of constructive interference between X-rays. All the gray areas with no spots are places where X-rays destructively interfere and they cancel each other out. So these patterns are due to the arrangement of the atoms in the crystal. So we can go work backwards from these patterns to determine the atomic structure. Here's what a data collection actually looks like. And this is very close to actually real-time data collection. This is a set of diffraction patterns obtained from a protein crystal. Proteins are huge biological molecules that typically have thousands of atoms in the unit cell making up the crystal. Data collection is extremely fast. On our CMCF ID beamline with the c.l.s, an entire data set with hundreds of images can be collected in less than 30 seconds. The whole cycle, including mounting and dismounting the crystal using a robot, takes less than 3 minutes. And you can operate these beamlines from home. Over 90% of our users operate the beamlines remotely. The users ship us the samples, we load the samples for their beam time, and the user takes over remotely using a robot to actually mount the samples. Here's an example of a protein structure or a large biological molecule, which was determined using single crystal diffraction. So protein crystallography is one of the most indispensable synchrotron techniques because solving structures for large molecules with thousands of atoms is often not possible using laboratory techniques. When you look at the structure, you might think, I don't see many atoms, but lots of ribbons and spiral structures. Protein structures tend to be so big with so many atoms that they're illustrated with idealized representations like this. Each of these spiral helix structures is actually made up of many atoms, but representing them with ribbons makes it easier to visualize the structure so that you can actually see the forest through the trees. Structural biologists know what these different ribbons represent. And a single view of this protein molecule doesn't really do it justice, but rotating it gives you a better idea of the full 3D geometry. So this molecule is a human protein complex from a study on the design of anti-cancer drugs. And the entire molecule is made up of over 10,000 atoms. Solving a structure like this would be very difficult, if not impossible, without using the synchrotron. My colleagues, Dennis and Kurt, run huge volumes of protein samples for industrial clients on both of the CMCF beamlines. This work is a critical component of our industrial science, or our industrial revenue, and it results in some high impact publications as well. So the molecule shown here is a COVID protein complexed with an experimental pharmaceutical. The two blue circles show where this pharmaceutical binds to the COVID protein. This pharmaceutical is an antiviral medication, and it's been proven effective against multiple viruses, and it's being tested as a potential treatment for COVID. CMCF is also very good for small molecule single crystal XRD. When we say small molecules, we mean structures that are not really huge biological molecules with thousands of atoms, but these structures can still be pretty big. So this structure is small enough that you can see all of the individual atoms. It's what is called a metal organic polyhedra, or a MOP. It's like a molecular cage that can be used for applications like drug delivery. So by changing the way different molecules are situated on the surface of the MOP, you can control how these structures interact with water, like how soluble they are. So by engineering the surface chemistry, you can tailor how these structures behave in water, and by extension, how they might function in the body. We talked about unit cells and how the unit cell is the brick you use to build a crystal. Here's what one of these MOP cages looks like in the unit cell used to build the crystal. This unit cell is a cube with an edge length of roughly 3.5 nanometers. That's roughly 10 times longer, or 1,000 times more volume than the diamond unit cell we looked at previously. And here's the entire unit cell as part of the larger crystal. So there are so many atoms in this structure, it's hard to see the forest through the trees. That's why it's easier looking at a single cage molecule, like the top left figure. I was going to show you the previous protein examples within a section of the crystal, but they are so big, it's really hard to actually see anything. So the previous three examples illustrated structures solved from single crystal diffraction. But sometimes you don't have a large single crystal. You just have a fine powder with millions of tiny crystals. But we can perform diffraction on powders as well. Instead of a loop with a single crystal on our sample base, for powder diffraction, we have a small capillary tube that's filled with powder. This capillary at the top right is about 0.5 millimeter in diameter, and you can actually see some of the powder on the outside surface of the capillary. When we get diffraction from millions of crystals, instead of a pattern of spots on our detector, we get a pattern of rings because all of the different possible crystal orientations get merged together into one image. Looks something like this. If you draw a red line through these rings, like the red line in the cartoon detector here at the bottom middle, you obtain a series of peaks that look something like this. You'll notice this 2D cartoon ring pattern has 4 rings, and the 1D plot beside it has 4 peaks, one for each ring. These peaks are like a fingerprint for the different crystalline materials that are present in the sample. A good way to think about a powder diffraction pattern is that it is a fingerprint for the crystalline compounds in the sample. Every person has a unique fingerprint, and every powder sample has a fingerprint based on the different crystalline materials that are present. Here's a powder diffraction pattern from a mineralogical sample. I look at lots of mineralogical samples, everything from geological exploration, looking for new mineral deposits, to monitoring mine tailings to help mitigate their environmental impacts. Mineral samples are often really complex with many different compounds present. In this specific sample, we can identify 13 different minerals using this data. Each of these minerals has characteristic peaks we can use to identify them. I've tried to use different colored sticks to illustrate each mineral in this sample. It's a little bit difficult to make them look distinct when you have this many different components. The largest peak and all the additional red peaks in this pattern are due to quartz. So quartz is one of the most common minerals in the Earth's crust, and it's made up of silicon and oxygen atoms. I've shown the crystal structure for quartz here on the bottom, or the top right rather, and the unit cell that serves as the building block to build the crystal. If we change the orientation of that unit cell to look down one axis, we can see that every silicon atom is bonded or attached to 4 oxygen atoms, and every oxygen atom is bonded to 2 silicon atoms. And so these atoms make a 3-dimensional network to help form this crystal. Here's another mineral. The olive green peaks in this pattern are due to hematite, which is an iron oxide mineral. And hematite is one of the compounds that makes up rust that occurs when iron metal gets wet. So if you think of the orange flaky rust on a rusty nail, part of that rust is probably hematite. Each of the minerals in the sample have a different set of peaks in the powder pattern, which you can use to identify them, and a different crystal structure or arrangement for atoms. We can use the powder pattern to quantify the different minerals or crystalline materials that are present in the sample, as well as just identify them. And there are different ways of quantifying how much of each mineral is present in the sample using powder data. So we won't go into the details of doing this. In this specific sample, the largest components are the quartz, which we looked at previously, and a mineral called nontronite. The broad peak at low angle in this pattern is due to nontronite, which is a type of clay mineral with a very small crystallite size. And that's why this peak is so broad. So powder diffraction is good at looking at mixtures of crystalline materials, but you can look at pure materials too. Most crystal structures are solved using single crystal methods, like I showed you previously, but you can solve crystal structures with powders as well. And for many technological materials, large single crystals can't be synthesized. So powder diffraction can be critical because there can be sometimes there's no other way to actually get to the crystal structure. This powder pattern was obtained from a new organogel material that could potentially be used for drug delivery or possibly even as a food additive. Has a very long, skinny unit cell, which is illustrated by the black rectangle in the crystal structure here. And analyzing this data was very tricky. I would hesitate to say that you couldn't solve this structure with laboratory powder data, but it was hard enough using the synchrotron data that I don't think I would want to try it with lab data. It was that difficult. The high intensity of the synchrotron allows you to look at very small sample volumes. So many art and cultural heritage samples are really small to minimize alterations to works of art. This is a paint sample from a piece of artwork. It's roughly 200 micrometers or 0.2 millimeters in size. And getting decent data in a reasonable amount of time would be tough for many laboratory diffractometers. The data here was collected in a few minutes using our CMCF ID beamline. And this paint has a mineral called anglesite, which contains lead, and another mineral called cinnabar, which contains mercury. So this paint is fairly toxic. Knowing the ingredients in the paint helps us to understand how best to conserve the painting, and it also helps us to protect the people that are working to conserve the painting. Synchrotrons really excel for time-resolved studies because of the speed with which you can collect data. You can track changes which are challenging or impossible to follow with the intensity and time resolution of lab instruments. High energy is also very important because low energy X-rays don't travel far in dense materials. With most lab XRD systems, you can't perform time-resolved work in transmission because the X-rays aren't sufficiently penetrating to actually go through systems like battery systems. But with synchrotron X-rays at really high energy, you can perform transmission measurements on real battery systems, like the battery pouch cell that's shown here. You can measure how peaks shift during battery cycling to better understand how the materials are behaving while the battery is actually operating. So we're going to switch now to our second area, and this is called X-ray imaging. Specifically, we're going to start with a technique called X-ray fluorescence mapping. X-ray fluorescence is a technique we use to identify different elements in our sample. We use an incident beam of X-rays that is high enough energy to excite different species of atoms in our samples when they absorb these X-rays. When the atoms get excited, they emit new X-rays, but the energies of the new X-rays depend on the type of atoms that emit them. So you can use these emitted X-rays to identify the atoms or elements that are present in your samples. An experiment typically looks something like this. We use mirrors called KB mirrors to focus the incident X-ray beam down to a very small spot on the sample. When the X-rays hit the sample, they excite atoms in the sample by knocking an electron out of the atom. This excited atom is not stable, and as the atom relaxes, it emits a new X-ray. This is called X-ray fluorescence. We use a detector to detect the fluorescent X-rays, and the X-rays from different elements will have different energies, so we can tell them apart. So this cartoon XRF spectrum at the bottom right here has peaks from 3 different elements: silicon, iron, and copper. But they all have different energies, and so they can all be readily identified. When we looked at powder diffraction patterns, we talked about how a good way to think of a powder pattern is that it represents a fingerprint of the different crystalline compounds in the sample. An X-ray fluorescence pattern is like a fingerprint for the elements in the sample. The different peaks in the pattern correspond to different elements. So this is an XRF pattern for an environmental sample. The largest red peaks in this pattern are due to iron, and there's a fairly large peak for zinc, shown in purple. And then there are some smaller peaks for other elements like calcium and titanium and copper and arsenic. So this pattern is like a fingerprint for all the elements in our sample. One of the main strengths of the synchrotron is the high intensity, which allows us to collect data really quickly. So if you use a very small beam, you can move your sample horizontally and vertically in steps in the beam to obtain the elemental composition at every point. And you can use this to create really detailed 2D maps. So this is a 2D map of the iron and zinc contents in the root nodules of a soybean plant. These nodules have vasculature on the outside, almost like veins. And you can see the iron in red is pretty evenly distributed throughout the nodule, but the zinc in green is mainly in the vasculature on the outside. XRF mapping is excellent for looking at distributions of nutrients or toxic elements in plants and animals. This is a study looking at the distribution of nutrients in oats. If you don't know what all the different features of the oats are on the top right, don't worry, I don't know them all either. There's a couple of different ways this data can be illustrated. You can make individual maps of different elements by themselves, as I've illustrated here on the left. But often we want to see how different elements are associated with one another on a single map. And this is what's called a tricolor plot, with 3 different elements on one map. So you can see here the manganese and the zinc form layers on the outside of the oat, while the sulfur is spread throughout the middle of the oat. And the embryo at the bottom right contains a lot of zinc, which is kind of highlighted by the blue here. Elemental mapping can be very powerful for understanding the effects of toxic elements. Mercury is one of the most toxic pollutants in living organisms, and exposure to mercury, unfortunately, is extremely widespread. So small fish called zebrafish get used for toxicological studies because they have a physiology that's very similar to mammals. So they're a really decent model for how toxins might behave in us. Here are multiple elemental maps, including mercury for a zebrafish. And you can see immediately that the elements are not evenly distributed. If we zoom in on the eyes and brain, you can see mercury is highly concentrated in the eyes. If we zoom in further still, the mercury appears to concentrate in a layer underneath the cornea and the epithelium in the eyes. So understanding how elements like mercury distribute themselves in the body can be really important for understanding their toxic effects. Here's an example of a post-mortem section of a human brain. And this was taken from a person who was suffering from multiple sclerosis, or MS. An iron accumulation in the brain has been observed in MS patients, and it's believed to be associated with inflammation and vascular abnormalities in the brain. The small maps here illustrate iron and zinc and copper in this brain section, while the large map combines all three elements. And you can see very intense hotspots with significant iron accumulation in this brain section. And the zinc and the copper, they're much more evenly distributed throughout the brain. XRF mapping has applications in cultural heritage preservation. Daguerreotypes were the first commercial form of photography in the 19th century. And so these photographs are made of sheets of silver plated copper, and different chemicals can be used to fix the images to the sheets. The problem with these photos is that the silver tarnishes or corrodes over time, and this can obscure the photograph. But we can use XRF mapping to see the original image using X-rays by targeting elements in the chemicals that were used to fix the image. In this case, they used fluorescence from mercury atoms to see the image underneath the tarnish. So the mercury XRF map pretty faithfully replicates the original photograph. Sometimes it's actually helpful to try fluorescence from different elements to see which element best reproduces the original image. In this case, silver and mercury fluorescence were attempted on one beamline with kind of mixed results, but using a different wavelength of fluorescence for mercury on another beamline produces an improved result. And they also tried gold on a second beamline as well. And the gold image doesn't have really high contrast, but it actually looks quite good. And you can tell the bottom two maps were taken on a different beamline because they are slightly different size than the top two maps. The last technique we are going to look at is another X-ray imaging technique called computed tomography. And this is a technique that can produce both 2D and 3D images. Some people will be familiar with computed tomography if you've had a CT scan at a hospital. If you consider a familiar hospital CT system, the patient is held stationary while the radiation source moves around the patient. With the synchrotron, we can't move the source, so the sample is actually rotated in the beam. As you rotate the sample, many 2D images are taken, and each 2D image represents an X-ray image at a specific orientation. All these 2D images are used to create a 3D reconstruction of the internal structure of the sample. Synchrotron CT is much faster than lab CT, which makes it very useful for time-resolved experiments. And tunable energy means that you can generally get better contrast than lab CT. You can also image larger areas, and you can image at higher resolution than you can with lab CT. So with computed tomography, we obtain a complete 3D rendering of the object, which can be sectioned or segmented as you need in order to answer your different questions. In this case, we're looking at a battery pouch cell, similar to the battery we looked at initially when we were looking at structure at different scales. And synchrotron CT is versatile because with control of the energy and the types of detectors we use, you can design the experiments to probe different length scales to answer different questions. So the far left figure here illustrates low resolution experiments on the battery cell level. And many batteries contain a liquid called electrolyte, which transports charged atoms called ions in the battery during operation. At the top left, we have a new battery with limited voids in this electrolyte fluid. But at the bottom left, you can see depletion of this electrolyte after cycling the battery for a couple of years, and the growth of gas voids, which can cause a battery to fail. If we zoom in a bit, the two middle columns illustrate the damage the cycling causes at the electrode level, like thickening of these electrode layers and formation of voids or delamination of these electrode layers. Again, this type of damage can cause a battery to fail, and understanding how damage forms can help us figure out how to improve the batteries to make them last longer. On the far right, even higher resolution measurements illustrate differences in particle morphology and microcracking of individual particles within the electrode layer after cycling. And if you look really closely, a lot of the particles in the cycled battery at the bottom right here look kind of crushed. And this is because the cycling has caused a lot of expansion and contraction, which eventually damages these particles. And this again can cause a battery to lose, can cause it to lose capacity or fail in the long term. Synchrotron CT is very fast, so you can even perform time resolve studies. This is an example illustrating changes in the liquid electrolyte of a battery. And as you increase the voltage in the battery during cycling, the battery electrodes expand, and this causes motion in the electrolyte. Each individual CT scan making up this movie takes less than a minute to collect. And you can study other types of time-dependent behavior as well, as long as they occur slowly enough that you can actually capture them sufficiently. Computed tomography is very important for studying bone and skeletal issues. This was a study looking at the incorporation of trace elements like barium and strontium in bone. Barium and strontium are similar to calcium, so they can substitute for calcium in bone. And this makes them very useful for tracking changes in bone. In this case, they were looking at barium and strontium incorporation in rat leg bones, including mature rats, which are finished growing, and juvenile rats, which are still reaching full size. So the rats were given extra barium and strontium in their diets. For the mature rat bone at the top row, most of the barium and strontium are incorporated in what is called the growth plate, which I've illustrated here with the red arrows. For the juvenile rat bones in the bottom row, barium and strontium are almost everywhere because the rat is still growing. So this work shows the potential for using these trace elements as contrast agents, and you can use them to track changes in bone growth for pharmaceutical studies to see how drugs that are promoting bone growth work and compare them to placebos and things like that. CT imaging can be used for different types of samples. And here's an example looking at the structure of a plant. So X-ray imaging can be used to understand the role of a plant structure and chemistry for imparting resistance to disease. And this is an X-ray CT movie of a wheat spike sample, and it was used to study resistance to fusarium, a fungal disease in wheat. The light green segmented portions inside the plant are the pore space or the empty cavities in the plant. And the amount of pore space is correlated to the plant's resistance to fusarium because the fungus has trouble spreading through these pores. So different wheat varieties with more pore space are much more resistant to fusarium. So by studying this plant structure, you can actually understand which plants are going to be more resistant to the fungus. This is our last example for the day. And these researchers were studying differences in plant vascular systems in different members of the Asteraceae flowers family, which is commonly known as the daisy family. So this is a fly-through video from looking at the head of a Gerber daisy, and it's illustrating the internal structure of the plant, which was determined using computed tomography. I can't personally do justice to the various features of this plant with words, so I'm just going to let you watch.
Those are spectacular images. Were those taken at c.l.s?
This was taken at c.l.s, yes.
That is very
Yeah.
cool.
Yeah, it's a very, very cool movie. Yeah, this paper just came out in the summer.
I may have to go and read that paper.
Pretty neat stuff.
It is.
So hopefully I have convinced you that the synchrotron is a really powerful tool for looking at structure in many types of samples across a wide range of different disciplines. Synchrotrons can be crucial for solving certain types of problems that are very hard or even impossible to solve using standard laboratory techniques. And it's really important too that I emphasize this wasn't a complete list of things that you can do with a synchrotron. It's just an illustration of some of the possibilities. There are entire sets of techniques that we haven't looked at today that can be done at a synchrotron. So this is sort of an idea of the possibilities and some of the different ways you can use a synchrotron. I would really like to thank many, many colleagues at the c.l.s. who have contributed to this work, and also some of our clients and collaborators whose data has appeared here. And with that, I would really like to thank you for your time and attention. And I hope you learned something, and I hope you really enjoyed today's presentation.
Well, thank you so much for this, Joel. It's you've done a really fantastic job of explaining some of these quite complicated techniques in a way that I find both interesting with the examples, but also understandable as a person who doesn't have a deep science background. So I thank you very much for that. And of course, as a reminder that while this talk was designed for industry collaborators that you may have been talking to, for us, we're reaching out to educators. But you've worked with me and some of our educators and some of our student groups as well.
Yeah.
And do you use the same techniques when you work with them?
Yeah, we've done X-ray diffraction with student groups. Not very often. More often, I think it's been X-ray fluorescence and
True.
mapping type experiments. But yeah, certainly we have applied some of these techniques with student groups. And this presentation actually, it was designed for a lay audience.
Okay.
Yeah, I created this presentation to talk to people that don't necessarily have a scientific background. So it's a little bit challenging creating a presentation like this simply because you can't assume knowledge. You
Yes.
have to try and explain all the different concepts that you're going to present very, very thoroughly. So
Yeah,
it's a
100%.
different type of challenge than some presentations I make.
For sure. And I really appreciate some of the schematics and diagrams that you created to help us understand some of these concepts. That's really, really helpful.
Thank you.
Um,
No, I really appreciate it, Tracy.
yeah, no,